So it's a pleasure to be here. It's a little bit scary to have that camera, so hi YouTube. So what I'm going to do, just to briefly recap, actually what we're going to do is we're going to give a synchronized set of talks where I'm going to start off with a pure whiteboard talk introducing some of the thought process that we went through a couple of years ago in thinking about how to learn dynamics from single-cell snapshots. And I'll talk about the limits that we came up against in dynamic inference. And that then stimulated us to think about ways of overcoming those limits. And in the second part of the talk, Caleb will give a pure PowerPoint talk where he'll talk about how we've been thinking about beating the limits, or I should say how Caleb has been thinking about beating the limits of dynamic inference by integrating lineage tracing information into single-cell analysis. And so now I'm going to just switch to what I'm going to be talking about. And if I can get through all of this in the time that we have, I'm going to briefly race through an introduction, but try to land as quickly as possible into a problem statement, try to formulate mathematically what we mean by dynamic inference, discuss the limits on inference, discuss a solution. There's more than one as we'll discuss. And then reflect on how we can practically implement it using ideas from spectral graph theory, and then introduce a particular method which implements it, which is the population balance analysis, or PBA method. So that's the idea for today. And do feel free to interrupt me, and we'll just see how far we get. So I think that what I should probably do before starting in general is just to give some context of where the motivation for this comes from. So my lab, some of which, or actually many of which are here now. You can pick up your hands, right? There you go. If you want to ask any questions, you can ask them later. So my lab is generally interested in understanding several aspects of fate decision making in cells. And this is the notion that an undifferentiated cell in development, in homeostasis, in regeneration, can commit to one of a set of differentiated fate choices. And there are a number of levels at which to ask this question. The first one is that a notion that to go from an uncommitted state to a set of stable states requires an instability of some sort, where you have some underlying process which can reliably switch between different discrete predetermined modes. And understanding, trying to gain insight into what those circuits are is an interesting problem. Then there's the idea that there's multiple signaling pathways which feed into decision making, and just knowing what those pathways are and how they influence decision making seems like a very important problem and very practically useful. Then there's the notion that these signals, even though they're acting at a single cell level, are in fact there to coordinate behaviors across a population to achieve some sort of balance, either in order to pattern an embryo or in homeostasis to ensure that we get the right numbers of different cell types. And understanding those circuits and how we get homeostatic control or feedback control is another level of complexity. And each one of these questions really lives on a different level of hierarchy. So there's many different ways of addressing these questions. What is somewhat unifying as a tool for many of our approaches is the use of single-cell analysis. And the general idea is that the reason that single-cell analysis, and I'll focus specifically on single-cell RNA sequencing, is so useful, and I suspect that this will be very familiar to most of you here, is the fact that we can immediately learn a great deal of information, some of which seems extremely basic, in order to lay out a basic map on which to start to understand differentiation. So some of the types of information that we learn about are what we might call the end states of a decision-making process, right? So these are the mature cell types. We might also learn something about the hierarchical structure of a decision-making process. So if we're generating multiple cell types, is that done through a single decision-making process with a very flat hierarchy? Are there a set of bifurcations or trifurcations? Is it even sensible to think about differentiation as a tree? Or maybe there are situations where we have, say, basins and convergence of differentiation for multiple trajectories, and it's dangerous to impose the notion of a tree. So we can start to ask these questions in some sense in an unbiased manner with single-cell RNA sequencing. And then there's the notion that, of course, if there is a hierarchy, then there might be transitional states which are important to know. From all of this, we can, of course, if we have information at the level of the whole genome, we can start to pull out information on expressed regulators, which could be candidates to understand the control of a system. So this is very preliminary in terms of understanding mechanism, but provides us with a very useful starting point. So these could be transcription factors, receptors, and so on, that we can then go on and try to perturb. And the lab is really, in many respects, trying to map out these differentiation processes and then start to understand how they're controlled through combining these readouts with perturbations. So this provides some sort of a very, very general background. And now I suppose that I'd like to get onto the problem of dynamic inference. And the issue, the idea, I'm going to actually have a few key takeaways from this talk. And I'd like to just write them down right now. All right, so if I get through nothing else, hopefully these takeaways will, I'll manage to get through and unpack them. So the first one is that learning dynamics is not the same as learning a manifold. So I'm now borrowing here some technical language. The idea is that if we have a snapshot of cells, you can imagine a single cell data set being represented in some high dimensional space. This is gene 1, gene 2, gene 3, and of course, there's going to be additional axes. That we can imagine a single cell data set representing a point cloud, and a very typical challenge in data analysis is to learn some low dimensional structure, which could be nonlinear over this data. And that type of structure, you can imagine, say, representing it by a graph or in other ways. That structure could be parametrized as some sort of continuous manifold. And there is a difference between parametrizing a manifold and just describing some structure and describing dynamics which occur on that manifold. So that's just it's a subtle point. It seems possibly obvious. The reason to call this out is that there's over 75 different methods for learning dynamics, and many of them are in fact just about parametrizing manifolds and not really about learning dynamics. So the second point to make is that, and this is probably why, of course, manifold learning is really what people do, is that there is no unique solution to a dynamic process from a snapshot. So the idea of uniqueness is sort of a very classical concept in mathematics, which is the idea that given that I have a problem to solve, is there more than one solution? And the key takeaway is that for the exact same data set, one can essentially find infinitely many solutions for the dynamics that could generate that precise data set. So this is just a practical problem. And I realize, a type of dynamic. I realize that I haven't fully defined what the problem is yet mathematically, but in the spirit of giving the key takeaways first, I'm just going to jump to something that I think intuitively will make sense, and then we'll do this more properly. So then the third point I'd like to make is that, and this is a completely separate point, and hopefully will be fun to reflect on for some, will be that spectral graph theory makes it possible to link biophysical models of dynamics to graph-based methods for data analysis. And this is sort of an exciting idea, at least for us, because graph-based methods have become extremely powerful. And if you think about many of the methods that are being used today for single-cell analysis, they do rely in many ways on going through graphical-based representations, whether it's UMAP, which uses, say, a weighted k-nearest neighbor graph, or just k-nearest neighbor graphs as a form of representation, which are used for clustering, for force-directed layouts. Many of the dynamic inference methods, the first thing that they start to do is to make a graph and then say, look for spanning trees. Many of these approaches are starting purely from a heuristic perspective, that these are useful ways of approximating manifolds or learning manifolds. And in fact, one can then reflect on those methods and understand what kind of dynamic model are they in fact implicitly or explicitly trying to model in the steps that they take. Right, so there's a very particular decision that's being made in constructing these graph-based methods, and those have implications for what type of dynamics we think are going on, whether we know that or not. So that is really the takeaway. And I should also now finally say that this work, which I'm going to describe, is described in far more detail in a work that we published, is Caleb's paper in PNAS. In 2018. Okay. So now let's see. So that's the key takeaways. Now let's get on with the problem statement. And I think I'm going to have to make some space. I could go on maybe one more block. Okay. So the problem statement. All right. Now, I should say I'm going to get a little bit technical. And for those who find that this is too abstract, just close your eyes, get some sleep, and when Caleb gives his talk, it'll all make sense. Okay? There's some great data in there, and it's like beautiful plots and so on. Okay. So we have a cell transcriptome. A cell transcriptome can be thought of as, say, a point in some high-dimensional gene expression space, right? So this is a single cell, sampled transcriptome. And a data set might be thought of as, say, a collection of these. This is a data set. Okay? And what we are assuming is essentially the model, in a way, which I think we would all agree on, is that cells are not static. So we can imagine that what's actually going on is that we've captured a cell in the middle of a snapshot where it's undergoing some dynamics, which is dx by dt is changing as a function of time with some velocity in high-dimensional space. And the question is, if we sample cells from this data set, can we make some statement about this dynamics? And to formalize this, we can write down what is essentially purely a bookkeeping equation, which is the population balance equation. So I'm now going to write down the population balance equation. And this equation, in a sense, is a truism, right? It's not so much a model as just a statement about what is inevitable. And let us write that down. So so first of all, I think that, there's one key point that we're going to add over here, which is that we're going to just define that x is sampled from some density, so probability distribution or density, which is going to be C of x, and it could be evolving over time t. All right, so as we come to the system, we come to a tissue, say, and we just take a few cells out and we have a look at them. There was some underlying distribution of cell states. And we can assume that that distribution is fairly well populated. If we would go to multiple individuals and so to get an infinite number of cells, there would be a perfect distribution that exists. But we're only sampling some number of cells from there. That could be thousands, tens of thousands. You know, it depends. These numbers are going up now. So the population balance equation is a bookkeeping exercise, which is saying that the rate of change of this density over time, this being the partial derivative with respect to time, can be thought of as essentially cells flowing into a particular point of space, out of a particular point of space, cells dividing or being added to that point of space, or cells being lost from that point of space. So that's why it's a bookkeeping exercise, because it's just taking into account everything that could happen. So motion between regions of space is just a divergence of a flux. And the flux over here is going to be the velocity, which it's going to be the mean velocity at a particular point, x, times the density of cells at that point. And then we are going to have the fact that, as we said, cells could also change their number in a positive way, which would be sources. And again, this would be at some point in time. And they could also leave at some point in time. So this is the population balance equation. And the thing that's missing over here, strictly speaking, is the fact that we've described everything as purely local. So you can only increase or decrease in this equation by the fact that you either divide and the two sister cells that now are created have the same exact state as before. And we have a situation where you can move from one region of space to the other, which is giving us this velocity. And you only change your density if there's a divergence in the velocity. So in other words, more cells are leaving than arriving, or more cells are arriving than leaving, and you have a divergence. Now, of course, this might not be the most general formulation, because we could also have non-locality. So if a cell divides asymmetrically, the two daughter cells instantaneously move to two different regions of gene expression space where there were no cells before. So there's some approximation being made over here, but it's a small approximation. We know what it is, so we're going to just stick to this for now. Okay, so now there's a couple of things to say over here. As I said over here, this is the average velocity at x. This term, which is a source term, can be understood in the terms of cell division. This term, which is a sinc term, can be understood in terms of death or exit. If you are, for example, looking at a spatially confined compartment, and at a particular state cells can leave that compartment, then they are capable of exiting the system. Right? Now, there are other ways of understanding this. You can also think that the way that you're purifying cells for your experiment is throwing away cells in some way, which is effectively imposing either a source or an effective source, because cells suddenly appear in a particular region of gene expression space because you've thrown away everything that was before that point. Or there could be a sinc because you've just thrown out cells which, say, start to express some differentiation marker. So that would effectively act as a sinc. So there's also experimental considerations that come into the way that this equation manifests itself. But by and large, we can think about those things and take them into account. So now the inverse problem becomes that we are sampling cells from C. We know something about the evolution of C with time. We can see that it depends on the average velocity. And we would want I'll just use a different color for this. We would want to make some statement about, we would like to learn V. Right, we would like to learn something about the dynamics of this process. Yeah?
So are you still considering the cell cycle inside the differentiation process, or you assume that you already corrected that cell cycle effect and not just
So as far as cell cycle is concerned, if cell cycle is impinging on the state of the cell, which it would be, we haven't specified here that we're talking about RNA sequencing in particular, that would essentially just correspond to an actual velocity because the cells are moving through a state space. Now, there could be multiple directions that cells are moving in. So they could be progressing. This is a high-dimensional problem. So this velocity, there's no problem for a cell to have a velocity in the direction of differentiation and another, say, circular velocity in the direction of cell cycle. Right? Yeah. Okay, and as the cell divides, the density of states goes up, and then we have an effective source at the point of cell cycle, which corresponds to, say, cytokinesis. Okay? Yeah. Okay, good. So this is good. So now the reason this equation is interesting to sort of stare at for a bit is that it immediately teaches us that our task is fairly hopeless. So I'm going to just point out why that is. And I mean this in a fairly strong sense that it's not so much a practical challenge as actually a sort of a fundamental problem, which is that there's just a loss of information that is unrecoverable in the process of doing taking snapshots of cells. How am I doing for time? Okay, good. So the limits. So the next part I'm going to talk about so that was to just talk about 3 limits on inference. And some of these are going to be obvious, and some of these are a little bit subtle. All right. So first of all, we can see that we are sampling from a density C. So at best, we can learn something about the density C. But the density C only depends on the average velocity at a particular point. It doesn't actually depend on the dynamics of the cells themselves. It depends on their average dynamics. So the problem number 1, so let's just say problems, is that x sampled from C of x, but C of x depends only on V of x. So we cannot learn actual dynamics. And in particular, there's 2 types of violations that we need to consider here. All right. So there's two. I'm going to put this types because there's actually a continuum between these 2. There's 2 extremes of violations, or I should say, rather, deviations of V from the average V. So the first one is what we might just call stochastic noise. So short-term fast fluctuations, if I take 2 cells in the identical molecular state and ask what they do, there is going to be some sort of stochastic memoryless process by which some of them go in one direction and other ones go in another. All right, so memoryless fluctuations. It's a noise, right? All right, so you could imagine, and then the second one.
Yeah, well, should we think of that as just something about the limits of the resolution at which we can measure a deterministic system?
No, actually, no.
It's like some quantum thing.
Let me write down the second one and then address that, because it's the limits of resolution could be the second one as well. So let me get there, okay? So the second one is the fact that there could be aspects of the system that we are not measuring because we say if we're doing a single-cell RNA sequencing experiment, we're still only looking at the transcriptome. We're not looking at chromatin state. We're not looking at proteins, phosphostates of proteins. We don't know something. We may not know about the spatial context of the cell, which may have a hidden variable, which will only manifest at some later time. So two cells could be in different parts of the embryo in identical conditions, but later on, one of those states will suddenly start to express Notch ligand, and the other one won't. So in that case, this is a hidden variable. We will not be able to learn it from here. Again, some of these things seem obvious. They're useful to just call out. So hidden variables. And now I'm just going to address your question. So hidden variables, which are what we might call slow fluctuations, because they don't just act instantaneously on the state X, but they have some persistent long-term effect. A cell could appear identical now, but this hidden variable will manifest itself at some much later time. It's a slow fluctuation. So now, where does noise in measurement come in? Noise in measurement is throwing away information about the state of the cell. Effectively, it's creating hidden variables. Now, what that might mean is that those hidden variables are very short-lived. So in other words, that information which I've thrown out might give me a slightly better resolution about what you're doing now, but it's not really going to matter in another hour for what the cell does. On the other hand, it could well be that that hidden information could be very important in predicting what the cell is going to do later. And therefore, it could actually be a persistent loss of information, which is important. And where that sits really depends on what the nature of the information that you've thrown out is. Is it just short-term information, or is there some really meaningful long-term information there?
Would you agree it also depends on where this system falls in the range between a chaotic system and a sort of,
yeah, so
even if you measured it at sort of inconceivable amounts of resolution, had all the hidden variables, even then it's not clear that you could necessarily a priori predict future dynamics, unless you want to add some notions of biology and evolutionary constraint and the fact that we're here.
Right. Right. So the notion that systems might be chaotic, actually, you're right that that's not. In principle, you could imagine a system which is chaotic, but where there's no noise, and therefore with enough resolution, the average velocity will still tell you where you are. It's just that you would need to be able to distinguish differences in velocity at a very, very fine increment. So that's a somewhat different point from this, which is to say that in any system, you just have this loss of information, which is the difference between V and average V. So chaos, I think, is a different, yeah, you're right, that's a different point from here. And we're going to not be so fancy. So yeah, that's a fair point. So this is limitation number one, and actually sort of it breaks down into two types of limitations here. And effectively, a comment is that anybody doing dynamic inference immediately typically makes a statement that there's no hidden variables. Because if there are, you're sort of lost. You might have a single point, and there's actually two processes, one set of cells flowing in one direction, one set in the other, and you can't tell them apart, and you're lost. So hidden variables typically are something that we just all give up on straight away. So the second point I will now make, and there are just a couple more, is, let's see. Yeah, so it's even worse, right? So 2 is that actually C of x and t doesn't even depend on V, right? So it's not even, I mean, I lied when I said, well, it depends on V. It's not even, it's even worse than that. It depends on, if you look on the divergence of VC. And this is important because there are actually an infinite number of vector fields which will have the exact same divergence. All right? And specifically, that means that even if I could make a statement about the average velocity, I can only make it up until the correction of a divergence-free field, and that's basically measure 0 with respect to all of the possibilities for the dynamics. So specifically what this means is that, say, if, let's say, you had a solution V is a solution, right, to your dynamic problem, your dynamic inference, then also say V plus some other vector field, chi, is a solution, provided that we can find, say, chi could just be the curl of any arbitrary field. So any continuous, this is any continuous field. I think it has to be differentiable as well, okay? So essentially, we can basically just take our solution, just add any field to it, and that's also a solution. So that's sort of stuck. And specifically, to make this a little less abstract, let's just draw this diagrammatically. Let's say that you're modeling the dynamics of some on a manifold, and you say, oh, well, I think that there's a flow going this way, and now you're at this bump. You could say, well, the dynamics could be doing that, but the dynamics could also be doing that. And there's no way for us to ever decouple these. So anything that's a rotation cannot be identified from a snapshot. Okay? So we have that as a second problem. Okay, and then there's, and so those are some of the sort of the more severe limits. Now there's some very practical limits as well. You'll just have to read through some archaeological text here. So 3 is that we don't measure C of xt, right? We just sample. So there's a statistical problem, right? We actually don't even know this thing that we're trying to solve. You know, we're trying to learn V from C, but we actually don't even know C, right? The population balance equation wasn't written in terms of x's. It was written in terms of the underlying density. And then the fourth point is that the population balance equation is a high-dimensional partial differential equation. And we can't numerically solve partial differential equations in more than, say, 3 or 4 dimensions before any computer will start to break, right? Even if you just have every gene as on or off, by the time that you have, say, 10 genes, that's a sort of 1,000 different states to start to model, and you can imagine that that would just diverge as soon as you have more states. So we can't. So this is so numerics is hard. And then 5 is finally the population balance equation had unknown terms, which were these source terms and sink terms. All right, so basically we're in deep trouble. We take a snapshot. And just by flipping around the order of the flows, we could get flows in different directions, changing where the sources and sinks are. We don't know the noise in the system. We don't know if it's slow or fast. We don't know whether there's rotations in the system. And even if we made a guess at all of that, we would then have to still try to infer the dynamics from random sampling of an underlying distribution. All right, so obviously people have made a lot of progress in this. So some of this may feel like it's a little bit contrived. But the fact is that some of these limits are extremely hard limits, right? So obviously the problem of statistical inference is a little less severe. The problems of knowing your dynamics are more severe. I should mention over here that this really ingenious method, which is the Velocito method that was developed by Peter Karchenko, the RNA velocity method, and that overcomes some, but certainly not all of these limitations. So with Velocito, you're directly making a guess at the local velocity. You do not get around the fact that there might be fluctuations. You're still only looking at average velocities every point. So this is still a fundamental limit. You do get around the problem of rotations because you're directly measuring the velocity field. So you can basically just have one solution. You somewhat get around the source and sink problem because you, again, have a particular direction in flow. So you're not so sensitive to your knowledge of sources and sinks. OK. So now I'm going to just make I think we're running a bit short on time. So I'm going to make a very quick attempt at a solution. And hopefully I can get through the key points in a very short amount of time. So I'm going to accelerate slightly here. But really the key takeaways, I think I've now gotten at least to 2 of my key takeaways, which was that there's a big difference between knowing the structure and knowing the dynamics. and that there are fundamental limits, that there's essentially no unique solution. So I've made 2 of these. Let me see if I can get through the third. It would be really nice if I can. Yeah.
So you mentioned this RNA velocity thing. So it is a beautiful method, but there are a number of very strong assumptions behind this, like the rates of decay of things and splicing rates, things like that. But can you envision writing similar like Fokker-Planck equation here, but in the augmented space of spliced and unspliced numbers? So here you're
Right.
looking at
Yeah.
total RNA.
Yeah, that's a very good point. I agree. Yeah, you could probably do that. Yeah, that's a great point. And you can then impose an additional very strong constraint on the relationship between them. But there'll still be some variables in there. Right, yeah. OK, so I'm now going to try. So essentially, what we're going to do is we're going to say there's no unique solution. But just as a thought exercise, let's see what assumptions we would have to make to get one. OK, and we're going to just choose one particular form of a solution. And there would be many other ways of doing this. So you could imagine, say, building a big GRN and fitting your data to a GRN. That's constraining the solution because there's a particular architecture you've chosen, and that architecture is then going to limit what types of dynamics could explain the data. We're going to do it in a slightly different way. So I suppose we're going to do a solution, right? And the solution we're going to have is assume gradient-driven dynamics and Gaussian noise. And memorylessness, so Markovian dynamics. And I think that that's pretty much, I guess that we're going to, you'll see that this is, this is going to be even stronger. And all of this together means that we can think about the system as a Fokker-Planck equation. So this is one model, but we know exactly what the problems are. So the idea over here is that we're going to just take our average velocity, and we're going to write this down as the gradient of a potential field. And we're also going to add, we're going to say that there's also some noise in the dynamics, which we can think about as having some diffusion constant. And then this is just the formal implementation of Gaussian noise. Okay? And in principle, if you actually think about this as being gene expression space, the noise that you have in each direction will depend on the abundance of that gene and the burstiness of that gene and so on. You could imagine it's highly anisotropic and inhomogeneous. Every single point in space has a different diffusion. We're going to just completely throw that out for now. And again, this is emphasizing the fact that there are limits here. We don't necessarily believe this, but we're just going to make an assumption for now, which is we're going to assume that this is isotropic and homogeneous. In other words, it's constant. Okay? And when we do that, we now can take our population balance equation, and our population balance equation greatly simplifies to the point where it now has the following form. 0 equals half d grad C plus grad C grad F plus R plus minus R minus C minus some differential, some time derivative. And for the purpose of the time that we have left, I'm just going to think about steady state processes. So this is useful for thinking about tissue homeostasis. And if you're thinking about development or injury repair, you would have to worry about this being non-zero. But the correction actually leads to the same types of formalism. OK, so it turns out, and then now in this, I'm just very conscious of time, I'm going to skip. It turns out that it's useful to decouple this into 2 potentials. One of them, which it counters the diffusion, so we're going to call this a confinement. And one of them, which transports cells from sources to sinks, which is the transport potential. And very briefly, the confinement potential actually has a very simple form, which is, and we in fact don't need to deal with it explicitly anymore at all. Okay, And then the interesting part is the transport potential. And that's what's going to bring us to spectral graph theory in a second, which is that we have now 1 over C grad V is equal to R plus minus R minus. Okay, and this some of you might recognize as the Laplace-Beltrami operator. Okay, it's a differential operator which is essentially weighted by the local density of, in space. So now we have essentially a problem, which was we now need to solve this problem. And if we solve it, we get the potential. And if we get the potential, we can reconstruct a Markov process which predicts dynamics. And from there, we can get ideas such as pseudo-time or mean first passage time, fate probabilities at every point in space. Everything gets unlocked as soon as we have V. Okay, so we just need to solve this equation, and we're essentially, then from there on, we can turn the crank on some very well-known Fokker-Planck formalism. Yes?
Is Fokker-Planck an ansatz here, or
it's,
are you deriving it from the
So essentially, you can think about it either way. You can think that we're systematically going and making one assumption after the other, and Fokker-Planck falls out. You can also just say, let's just assume Fokker-Planck and then figure out what that means. And we actually understand every single assumption that required to go from the population balance equation to a Fokker-Planck equation. So there's no mystery in that connection. You know, it depends how you want to do it. It would be a little bit disingenuous to say we were surprised to get a Fokker-Planck equation. But it would be also wrong to say that we don't understand. It's not a pure ansatz. In an ansatz case, you're just really sort of guessing that this would be a good solution. In this case, we can tell you exactly what mistakes we're making. And hopefully in the next session, Caleb will show you that we can go back one at a time and test the assumptions and ask whether we're satisfying those assumptions or not.
So in the case where you just had a cyclical flow, right?
Yeah.
So where does that disappear?
Yeah, where does that disappear? As soon as you claim that velocity is a potential gradient, there's no way of hosting an oscillation because you can only roll down a hill, right? There's no inertia, right? So it's just like dropping a ball through honey, right? It'll never go around the funnel, you know, and down there, if you've been to the Children's Museum, right, where you throw a ball and it goes around. And it's never going to do that here, right? It's just going to roll down the side of the hill and get to the bottom.
Is that what you mean by gradient-driven dynamics?
Yes, that's what we mean. It's an overdamped system, so this would be the equivalent of rolling a ball down a gradient in honey, right? There's no inertia. You're just responding to the local potential gradient. And you can think about a gene regulatory network as implementing a gradient. And this has been very popular in the context of thinking about Waddington landscapes. A very important point to say is, if you write down an arbitrary gene regulatory network, it will not satisfy gradient. So Waddington landscapes don't describe most of reality. Okay, there's many, many ways of creating. In fact, it's very hard to create a gene regulatory network which has a Waddington landscape underlying it. Okay, most of the time, you have some sort of oscillations or structured rotational motions in space which cannot be described in this way. So it's a very strong constraint. Yeah.
But is there separation?
Go ahead.
Is there separation of timescales in which that this sort of emerges naturally from like kinetic equations?
It could be that there are certain systems in which it's possible and justifiable to ignore rotations. It could be that there are others which you can't. Yeah.
So Fokker-Planck and the standard sort of diffusion maps that people use are very, I mean, essentially they're just different powers on the density normalization.
Right.
So are you able to, I mean, would you say the theory you're developing here is sort of its own beast, or are there ways of kind of transporting between?
Let me link. So, okay, good. So that's now actually the final part. So at this point, we have this equation, and now we want to make contact with our data. And what we don't want to do is to numerically solve some partial differential equation in high-dimensional space. That's never going to work, right? So now there's a very beautiful set of results which make the link between diffusion maps, UMAP, you know, KNN graphs, and these types of equations, and show that the way that we choose to represent our data has different implications for how we're choosing sources and sinks, or how we're choosing to think about the dynamics of our system. And this is now going to be the final part, probably, and then I'll leave because I'm already over. So this is spectral graph theory.
You're okay. You can take 5 more minutes.
Okay, awesome. Well, yeah, okay, good. So essentially, we have a point cloud. Just to remind us, which was x1 to xn, and we construct a graph with adjacency matrix. Rho normalized, of the following form. So we have Aij, and we're going to have some sort of kernel, which is going to just tell us what the weight of the edge is. This could be 0. And then we're just going to normalize this, row normalize it, xi, xk. OK? All right, so that's basically just the starting point for defining a graph. And now a random walk graph Laplacian can be thought of as the identity matrix minus the row normalized adjacency matrix. And this is sort of the spectral graph theory 101. So there's this beautiful result, which is quite recent. And Caleb burst into my office with this result, and he was excited a few years ago, and this was just really electrifying for us, which is attributable to Michael Jordan's group in Berkeley. So this is Ting, Huang, and Jordan. And this is ICML. This is published ICML 2010. Okay, it's on Archive, I think, and you can find it in a couple of other places. And the result is as follows: so let me say sampled. Let me just add over here sampled from C of x. It almost looks like they knew what we wanted to do, and they showed that if you take the limit of sampling to infinity, so this is a asymptotic result, we're sampling from C, that this graph Laplacian converges onto a family of differential, second-order differential operators. Sorry. And which has some very generic form. And the precise values of sigma and mu depend on the choice of the kernel. And that was a really important result. The reason this is so important is that this now gives hope. So I'll tell you, this by itself is not enough. You need to know how to figure out what sigma and mu are. But the reason this is so important is if you look at this equation, you can expand it out and see that it, of course, has precisely this form, where the way to think about that form is that we have, so 1 over c, so example 1 over c grad is equivalent to having sigma equals 2 and mu equals 1 over c grad c. All right, so you can expand it out, and it turns out, so now we have a pretty simple challenge. If we can just figure out the right graph, we can, how to construct our graph from our data, we can actually approximate the graph Laplacian operator that we need in order to solve our problem. And at that point, we just have a simple matrix inversion problem, because we've just written this down in terms of a graph adjacency matrix. We just can invert the graph adjacency matrix and get V. So, that's really the genius of this. And it turns out that there's a very precise form. So I'm not going to write down the full form of sigma. Oh, I guess I do need to say one thing. So they solved this for a very particular case, which is where you have a k of xy, which is equal to some weight function, which depends on xy in a very general sense, and then a top hat function, so it's either 0 or 1, which depends on the distance divided by some spatially varying distance. And this is an extremely general form, because there's no statements about R or W except that they're differentiable. So you can essentially have some sort of very complex kernel. And again, this covers UMAP, this covers KNN graphs, and so on and so forth. Okay? So the key point is that they showed how to relate W and H, which is your, or W and R, which is your choice of graph construction, to the particular Laplacian operator that you're going to get. And it turns out that from this, you can show the two important results. So the first one is unweighted, so an unweighted KNN graph in high dimensions gives LRW equals 1 over C grad C grad. All right, so basically all we need to do is to construct a k-nearest neighbor graph, and we have our solution, right? Just like that. And that's a very powerful result that overcomes all the problems of numerical integration and so on. And the second result, which is, and I'll end there, is that the Markov matrix the Markov sort of transition matrix, right? So transition matrix can similarly be written down as is a KNN graph. So it's a KNN graph with weights W x comma y equals e to the minus v of y minus v of x over d, which is our diffusion constant from before. So we now have, apologies, that's like way back down the corner. We now have essentially the two components that we need to solve our problem. We have a way of turning this partial differential equation into a matrix equation, which directly gives us v, okay? And then the second point that we have is that once we know v, we can go and construct a Markov matrix which will recreate all of the dynamics on the system. And from there we can calculate fate probabilities, we can calculate mean first passage times, and obtain all of our dynamics. So now, of course, all of this is done with respect to extremely strong constraints on the dynamics. But the key thing is we've at least gone into this process knowing the ways that we could be wrong. So just to reflect now, if you think about, as I said, there's this paper now out which compares different trajectory inference methods, and there's over 75 of them being compared. Most of them will just choose some heuristic in which they're parametrizing space. And it would be interesting to know, at the end of the day, each one is making a particular choice. How many of them are differing just in their choice of statistics, but they asymptotically converge onto the same behavior? And how many of them are fundamentally predicting different types of dynamics because the underlying assumptions about the noise in the process or about maybe even they're not even conserving flux, right? Some of them may be throwing away flux and so on and so forth. So this is probably more philosophical than practical in identifying that there's a way of going back and reflecting on what are the assumptions that are being made in a particular process. And of course, it results in a practical pipeline, which is then implemented through this PBA method, which is available on GitHub. So I won't say any more about that. OK, I think in the spirit of time and moving on to some data, and I think Caleb will also review this pipeline sort of graphically, we can stop now. Thanks very much.
OK, well, hello, everyone. I'm Alon's disciple, and as such, I'm concerned with the question that he posed at the beginning, which is how do stem cells make decisions? And in particular, I've spent 4 years obsessing about the hematopoietic system. So it's really remarkable. If you take a moment of silence and listen to your bones right now, something incredible is happening inside of them. In a single day, over, 100 billion platelets are made, and 10 billion granulocytes. This thing is pumping out cells unbelievably, and that process stems from a collection of stem cells that reside in the bone marrow and self-renew and also differentiate. And people have, since the identification of these cells, there's been a long effort to understand how this process occurs. And recently, with the advent of FACS and cell sorting, people have arrived at a kind of canonical model. So if you open a textbook about hematopoiesis, you'll probably see a diagram that looks like this. So the basic experimental toolkit that people have used to arrive at this is to sort cells based on surface markers and then differentiate the cells in vitro or to transplant them into a mouse and then see what they do. And on the basis of this type of experiment, people have constructed a tree-like model in which stem cells in the bone marrow first make a decision between myeloid fates or lymphoid fates, And then, upon having made the first decision, they make a sequence of subsequent decisions until the final decision is made and they become committed to a single fate. And the connection between this experimental method and this model is that each one of these cell types was discovered as some sort of FACS gate. And then, so for example, common myeloid progenitor was found that if you take this FACS gate and you differentiate it, you get this collection of cell types. Whereas if you take this FACS gate and differentiate it, you only get this subset. So this model is a workhorse. It underlies a huge amount of our understanding of hematological diseases, of leukemias. However, it's not the final story. And one reason we can start to question this picture is that it's undergone so many revisions since it was originally published. So these are all just screenshots from studies that have been published since the sort of formation of the canonical model. And you can just take my word that all of these trees are slightly different from each other. And just to go into a little bit of detail about the nature of the differences. So this was one of the sort of major revisions of the model. So on the left is the canonical model. There's a decision for lymphoid cells on the left or myeloid cells on the right. Then what happened is that people discovered a cell type that they call LMPP, which is a lymphoid and myeloid progenitor that can make lymphoid cells and the GMP node that's in the middle in the right-hand tree, but it can't make MEP on the right. And so not only is this like a structural revision to what we saw on the left, but actually it's almost a paradigm shifting revision because it says that it's not actually a tree after all, that there's two different roots to the same final cell type. Another type of revision that's occurred is in the point where fate commitment actually happens. So again, on the left, I'm showing the classical model, and on the right, this figure shows the revision that was arrived at in this study, in which the tree is like unzipped. So now, instead of a single common myeloid progenitor, this cell type has been split into actually 4 cell types, which are all immature but committed progenitors of the final fates on the right. So now we're left with the question, this model keeps changing and changing, so what's up with that? How can we explain that? And one insight that's come more recently has been generated by single-cell RNA sequencing, and that's the realization that these cell types that are part of the canonical model are actually heterogeneous in their state. So this is a study from 2015 from Ido Amit's lab, where they systematically did single-cell RNA-seq on the whole hematopoietic hierarchy, and they showed that many of these canonical cell types are in fact consist of cells that are already transcriptionally primed toward different states. So with the advent of single-cell RNA-seq, people are starting to move from a discrete model to a continuum model. And I wanted to offer this diagram as a way to reconcile all the contradictions that we saw previously in terms of the continuum. On the left here is a discrete model similar to the ones that we've been seeing before. Here is what I propose, and it's not me who proposed it, other people did, what's actually going on. Actually, there's a continuum, just a sort of continuous landscape of transcriptional states, and on that continuous landscape, a graded process of fate commitment. And what we were previously conceptualizing as discrete cell types are actually just different ways of carving up the continuum. And you can imagine that if you took that continuum and you carved it up a different way, you could get a different tree. So that's why, so we can now reflect on all the different trees I showed you as just different ways of carving up the continuum. And that all the confusion is because we're trying to take a continuous process and force it onto a discrete model. So luckily now, thanks to Alon and other people in the field of single-cell RNA-seq and other single-cell like omics methods, we have a way to directly access the continuum, and that's what's being shown on the right. However, here we hit a snag, which is that unlike with FACS, where we had the ability to actually take the cell types and differentiate them and see what they turn into, now we can't do that. One is computational, and it's a recap of what Alon just talked about, and the other is experimental based on lineage tracing. So first, computational. So Alon just described the thought process and the mechanics of PBA, and I'm going to show it to you actually implemented on a data set. This project is a collaboration between Sam, who is my colleague, who's sitting in the back, and Marav, who's a professor at UMass, and Alon, and Betsabe, who's a student of Marav's, or a postdoc. And all right, so here's the project. We used InDrops to profile 5,000 KIT-positive hematopoietic progenitor cells. KIT is a marker of being a progenitor. And on the right is a low-dimensional visualization of that process. To orient you on this figure, where it says MPP, that's the starting state. That's where the stem cells live and regenerate. And then they differentiate out on the different arms of that diagram. And the tips of that diagram is where cells turn off KIT. So they just leave our data set because they're not being sorted for anymore. If you zoom in on the region around the progenitors, we can see a little more detail of the landscape that the cells are moving along. So one immediate takeaway is that, as has been pointed out by others, it doesn't look very discrete. It's just a big blob. And that might be because we're not representing it very well, or it may indeed be because, but for now, this is the best that we can do the low-dimensional visualization, and we're thinking about it as a continuum. And what we want to do is learn some dynamics on this, have some way of mapping cell fate onto this continuum. And to that end, we're going to use the principle of population balance. Here's the graphical flowchart that Alon promised of how we're going to apply PBA. So at the beginning, we have, I'm not allowed to point, sorry. At the beginning, we have a bunch of dots. Those dots is our single cells. And we want to infer dynamics. So we're going to apply the population balance law. That's the box, second column, first row. And then we're going to have some assumptions. First, we're going to assume no hidden variables. That means that everything's Markovian, and we can use a Fokker-Planck equation. And then also, in applying that Fokker-Planck equation, we're going to assume a potential or Waddington landscape because we can't do any better. So it's like, if you don't know something, just assume it's 0. And then we're going to input some parameters. This is R, the sources and sinks, and D, which is the degree of stochasticity in the system. And then also, we need to know the rate of change in cell density. But luckily, we're in a homeostatic system, so that's 0, unless the mouse is like stressed out or sick. And OK, so from there, we're going to solve the Fokker-Planck equation using the Ting-Huang-Jordan theorem. And then we're going to infer the temporal ordering of cells and the fate probabilities of cells. So in terms of actually implementing this method, we need to, so some of these assumptions, they're just automatic. They're baked into the method. So no oscillations. There's nothing to decide there. We just are setting it to 0. But for the diffusion and the sources and sinks, those are actually numbers that we have to come up with and input into the model. So with so we're going to use some heuristics, basically, to figure out what these are. For diffusion, we're going to use the fact that if the diffusion is higher, that means that cells can move around more, and that means that the number of cells which have the potential to go into each fate is greater. What that means is that for any given rate of diffusion, we have some sort of implication for the number of multipotent cells in the system. And by using prior knowledge on the number of multipotent cells, we're going to decide on diffusion. Then for the rate of birth and death, here it's really complicated because this is a really complex system with different proliferation rates in different parts of the manifold. We're going to just elide over all of that complexity and say that, within an order of magnitude, all the proliferation rates are roughly the same. They're high. The system, as we said, you're making 100 billion cells a day. And we're just going to assume it's, and also there's experimental data to show that on different parts of this landscape, it's within roughly 2 to 3-fold or 5-fold of each other. So we're going to assume positive proliferation everywhere on this structure, except at the endpoints where our selection marker is being turned off, and there cells are flowing out. So wherever you see red on the right, that's negative, that's a sink, and wherever you see gray, that's positive, that's a source. So here's the implementation of the method. On the left is the manifold we started out with. Then the second version of this diagram shows the transport potential, V. This is like the Waddington landscape that gets cells to move from one place to another. So blue means high up on the potential, red means low down on the potential, and cells are being driven, according to our model, cells are flowing from blue toward red. And then on the right is the calculation of fate potential based on, as the final output of the model. So for example, you can see that red used to live at the bottom left arm. I'm just gonna do it here. Whereas now the red is going all the way up into the middle. So we've sort of inferred which cells more toward the middle of the diagram are fated to go to the erythroid branch. All right, so we just did that, and okay. So now let's revisit our original motivation for doing this whole thing. The reason we went into this is because we want to try to explain the different results that people have gotten through facts as a continuum. So the most basic criterion for us to believe what we did is that if we carve up the manifold in the same way that people have done through facts, and then we take the average fate probabilities in those regions, we should recapitulate what people found through facts. So that's what's being done in this figure. Each column here is a different facts gate. On the bottom, the red dots show where we mapped, where we inferred those cells that facts gate lives in our graph. And then above that is a comparison of the fate probabilities estimated by our method and found in the publications that identified these facts gates. And hopefully what you see here is that there's a rough correspondence. And then we can quantify that on the right. So that scatter plot on the right shows on the x-axis the actual empirically measured fate probability on average for the facts gate. And on the y-axis is what we estimated. So, all right, that's that. So that's PBA. So when Alon and I got this result, we were like, oh my God, we did it. We solved biology. This is amazing. Best method ever. And so we don't want to blame Alon. I'll just say we submitted a paper brashly with the title, Asymptotically Exact Prediction of from high-dimensional snapshots. And we were like, oh yeah, this is like so cool. But then we completely got slammed. And in retrospect, and so this was the line. So reviewer 1, reviewer 2, we were like, oh, they just didn't get it, they can't. But then reviewer 3, they really got it. And then this was the line that we were like, oh wow, like we really messed up. Or we framed this wrong. So what reviewer 3 said is, thus, I think that claiming exact prediction is highly misleading. What is exact is not the prediction of biological behavior, but only the approximation of one model by another. Very true, right? So, like, we assumed no rotations, we assumed we knew sources and sinks, we assumed we knew the diffusion, we assumed all these things, like a preposterous level of assumption. Then we asymptotically, we don't even know where we are on the asymptote, we asymptotically solve the model conditional on all those assumptions. So this is far from an exact prediction of actual biological behavior. And on the basis of this humbling reviewer comment, we actually went completely in the other direction, and we just said, we were like, yeah, you're right. We can't figure it out, and actually no one can figure it out. It's impossible. So we were like, fundamental limits. That was the new theme of the paper. Okay, so now let me just review really quickly what the limits are, although Alon did a great job on this last time. So first of all, entry and exit points. So we have to know where cells are being added and where they're being taken away. I showed you before that we were just like, everywhere, they're proliferating equally everywhere except at the endpoints where they're leaving. That like we don't actually know that. That was just a heuristic guess. So this is something we just don't know. Then rotations. We assume they didn't happen. Then these two, now I just want to say that with, as Alon pointed out, with Velocito, you could kind of solve some of these. But here we have a little bit more of a profound limitation, which is the fact that even if we know average velocity, individual cells don't necessarily, their velocity isn't equal to the average velocity. And because this will be the focus for the rest of the talk, I'll just go into these in a little more detail. So the first uncertainty is hidden variables. So as Alon described, a hidden variable is something that determines what the cell is going to do over time, but we can't measure it. So that could be chromatin state, position in the embryo. It could also just be a measurement of the state that, it could just be that our measurements are sparse, so there's a low expressed gene that actually has a profound implication, but we're not measuring it, and thus that becomes a hidden variable. And another way to think about hidden variables is that a hidden variable means that the dynamics are not Markovian with respect to the measurements that we're making. So in a Markovian system, the definition of a Markovian system is that if you know the present state, then you can predict, then you know everything you need to know about the future. There's nothing, like any uncertainty you have about the future is because of inherent stochasticity, not because of some enduring feature that you didn't measure. So this assumption, hidden variables, the question is it Markovian or is it not Markovian with respect to our measurement? The second is stochasticity. So it's possible that cells, it's possible that you know everything, there's no hidden variable, but you still don't know exactly, individual cells can still be doing different things because gene expression is bursty because of, there's quantum fluctuations. And so that's the second fundamental unknown. Okay, so we just, all right, despite, even though we were happy about PBA, clearly it has its limits. It has fundamental limits. It has practical limits. And what we really want to do is we just actually want to measure the fate probability of cells and not infer it computationally. So now I'm going to transition to telling you about a experimental attempt at measuring the fate on transcriptional landscapes. And I just want to set up the questions that are motivating that effort. First, on the biology side, if we could really do this and accurately measure fate on transcriptional landscapes, we immediately our first priority is probably to just go answer a bunch of questions about hematopoiesis. So let's just revisit classic questions in hematopoiesis, like where does lineage commitment happen, what's the fate of transcriptionally prime states, but now through the new lens that we might have. Then after that, we can reflect on our computational inference. And in particular, we're going to ask three questions. One is, how accurate is PBA, now that if we have some sort of benchmark to compare it against? The second is, are there hidden variables? And then the last one is, let's actually measure how stochastic cell fate is. Okay, so this is what we would love to do. We want to be able to measure the state of a cell and then see what it does later. But we can't do that because with a single cell RNA-seq, we kill the cell in the process of measurement. However, something that we might be able to do is let a cell divide, then look at the state of one of the daughters, then let the other daughter differentiate, and then look at its state. And this will be a proxy for this, provided that the state of the two daughters was similar to each other when we measured the first one. And to the extent that those two daughters are different, that's going to define a kind of resolution. So if they're very different, that means that we have a very poor localization of the starting state. And if they're exactly the same, that means we have a perfect localization of the starting state. So that's going to be the fundamental constraint on how accurate our method is. And in fact, there's actually so with that in mind, there's three types of measurements we want to make. The first, and from here on out, I'm going to use the word state to refer to the state of the cell early on, like in the blob of the transcriptional landscape of progenitors, and I'm going to use fate to say what it did later. Obviously, these aren't fundamental, like, one woman's state or fate is another woman's state, because after the cell differentiates, into, say, a B cell or T cell, it may further differentiate into different types of T cells. There's nothing inherent about state and fate, but it's just with respect to a certain experiment or question, there's what happened before, and then you're interested in what happened after. So there's a state-fate coupling. That's how does the state at the beginning affect what happens later. The accuracy of that depends on the state-state coupling. In other words, how similar are cells at the early time point, or our sister cells. And then also, we're going to later, it's going to be important to ask how committed were cells when at the beginning. So that's going to be the fate-fate coupling. If we let two cells differentiate in parallel, how often do they do the same thing? And we want to set up an experiment that's going to be able to address all three of these questions. And that experiment is enabled by this barcoding tool that was developed by Alejo in the Camargo lab. And it's basically a lentivirus that delivers GFP with a random barcode in the 3' UTR. And so this is sort of like, this is like a single cell compatible lineage tracing tool. So the idea is that we just have cells, we use a lentivirus to put in this transgene, and then when a cell divides, both of the daughter cells get the same barcode, and then we can read the barcode when we do this single cell RNA-seq. And the particular experiment we're going to do is as follows. Take hematopoietic progenitor cells out of a mouse. Then we're going to barcode them. Then we're going to wait 2 days for them to divide. So now there's sister cells that share barcodes, and hopefully they're similar transcriptionally. Then we're going to just take half the cells out and measure them immediately. And the other half of the cells, we're going to let differentiate for 2 more days. Then we're going to take some more out. Then we're going to let them differentiate for 2 more days and then measure the rest. And sometimes barcodes will be split between here and here, and that's going to be a state-fate coupling. Sometimes two cells with the same barcode will both be sequenced in the first time point. That will be a fate-fate. And sometimes 2 barcodes. Oh, and another thing I forgot to mention is that the fact that there's 2 lines that go all the way to the right is because we're going to separate the cells also into 2 different wells, and that's going to allow us to see how committed cells were. So when a cell just divided and the 2 daughters get placed in separate wells, how often do they do the same thing? Here's the data set that we got. So my lab mates who don't study hematopoiesis call this alphabet soup. So it doesn't really matter what these names are unless you care. But the point is that it's 130,000 cells across 4 time points. Here's a low-dimensional visualization of what they looked like. And yeah. And so then we can ask where barcodes sit if we look across the time points. And the answer is that we have a good statistical sampling of the 3 categories that we care about. So we have more than 1,000 state barcode pairs, state-fate barcode pairs, and fate-fate barcode pairs. So hopefully we'll get a robust picture of what's going on. And to give you a sense of what the data looks like, on the right, I'm showing you examples of 3 different clones with different types of behaviors. So the gray dots is just the whole landscape. The colored dots is the members of that clone. And the color of the dot is what time point I saw the cell at. So for example, in the top picture, you can see that in this clone, the cells are just going in a straight line down one of the branches. In the second, you can see that over time, the clone is bifurcating into 2 branches. So whatever cell originally got this barcode was bipotent, or more than, it was at least bipotent. In the third example, that's a clone that's just staying roughly in the progenitor area over time. So that's some kind of self-renewing clone. So to get a systematic picture of the relationship between state and fate, our main criterion is that cells that at the beginning, at the first time point, sister cells are transcriptionally similar. And that's what's being addressed in this figure. So on the left, I'm showing examples of clones where we sampled the whole clones where I'm just showing you what happened on day 2, on the first time point where we did sequencing. And each color is a different clone, and what you can see is that the clones are relatively localized on the landscape.
A question about localization from the previous slide. So it seems like if I were just looking at this, like things are flowing, I'd be worried that like the one in the middle managed to split into two groups, but the one on top somehow managed to go straight down. And so like, to what extent do you think locality in these dimensional reduction plots has something to do with the actual geometry of how these things are able to move?
Yeah, that's a great question. So, I mean, you can almost break it down into two levels. There's, in the high dimensional landscape, how much are different types of behaviors actually localized to specific regions? And then when we look at it in 2 dimensions, how much do we preserve like a good partitioning between those relevant regions? And, you know, so the first question is kind of a question about hidden variables. It's asking, you know, are there unseen dimensions that are good at discriminating these different behaviors? And I'll have some slides later that address that. In terms of how good the low dimensional visualization is, I guess this, let me also get to that when I do hidden variables. Okay, so here we're back. So the question is, how similar are cells on day 2? This is giving us like the resolution. What's the resolution that we have in terms of connecting the fate of cells to what they do later on? And to address this systematically, we clustered the day 2 state space into clusters, so that's the colors in the middle, and then looked at the joint distribution. So on average, so when I take 2 different cells that share a barcode, which is the pair of clusters that they fall into? And the density along the diagonal says that like pretty often they fall into the same cluster.
What is the upper bound of the number of replications that can happen in 2 days in hematopoiesis? Like how many cell divisions are happening for?
So the population grows like 6-fold, but so there's a couple complications. One is that different cells are proliferating at different rates. And the other complication is that the integration of the lentiviral barcode happens asynchronously between 12 up to 24 hours or more. So the actual number of cell divisions that happens post-barcoding could be pretty variable between different clones. However, we don't see any real dependence of this level of similarity on the number of divisions.
At the beginning, you're sort of Poisson loading in some barcodes. You had to make a decision about how many barcodes. How did you think about that? Because there's trade-offs, right?
So more barcodes is always better, because as long as you can see all of them. Because it doesn't matter if 2 cells, so if one cell gets 2 different barcodes, that's now like a barcode squared. That's just like extra information on the identity of the cell. And in fact, there's another method called CellTag that was created by Samantha Morris that's very similar in its purpose to this. And there, each barcode is a pretty low diversity, but they have like a super ridiculously high titer. And so every cell gets like a bunch of barcodes. And so that's how they generate diversity. So more titer, more integrations is always better. But we have an operational question, which is that if we see 2 cells sharing, when we see 2 cells, do we call them a clone if they share one barcode or all their barcodes? And in our case, we just want to be conservative. Like it's just a loss of data if we fail to say that 2 cells are the same clone, but it's pretty catastrophic if a false positive is much worse than a false negative. So we just said 2 cells are only the same clone if they have all their barcodes in common. Okay, so anyway, the summary of this slide is that the cells are pretty similar on day 2. This clustering gives us an indication roughly of the resolution that we can trust. So now we're set up to answer, or to at least have some initial hints at the first question, which is, where does fate commitment happen on transcriptional landscapes? And so I'll just explain this figure. The gray dots is the whole data set. The circumscribed regions are areas that we called mature cells. So once a cell enters into one of these regions that's outlined in a certain color, that means that the cell officially, according to our definition, it officially differentiated into that lineage. And the colored dots are cells from day 2, which are being colored according to the dominant fate that attained by their sisters that were profiled at a later time point. So all the red cells are day 2 cells where they had a sister in a later time point that went into the red region. And so, yeah, we can learn a couple things from this. One is that there's like a salt and pepper pattern. So there's regions where cells can do multiple things. Another is that there appear to be sort of sharp boundaries. So there's an area where the red suddenly stops and there's only green. That indicates that there's some sort of change of gene expression at the boundary of the red where cells lose the capacity to become neutrophils. Yeah, I'm not going to dwell too much on this, but so that was the outcome of that. Next, transcriptional priming. I'm just rushing through the biology because at heart I just like math, so I'm just going to do the math part. But in this figure, I'm focusing on the progenitor cells, and what's being shown in the top row is the expression of known regulators for that lineage, and what's being shown on the bottom is the position of the putative progenitors that was identified through this assay for that lineage. And then hopefully what it looks like is some sort of co-localization in 2D. And that gives us some hint that these regulators, that like lineage transcriptional priming with these regulators has a functional implication in driving the cell toward that particular state. Okay, so that's it for biology. Okay, now we can go on to the math part. So first, PBA, how did it do? The answer is, as the theme of this talk is a little bit trashing PBA, but basically it didn't do very well, or in particular in the early states. So what's being shown here is just looking at a particular bifurcation between neutrophil and monocyte. And what's shown on the left diagram is the smoothened clonal fate bias that was identified through the lineage tracing. Then in the middle plot, I'm showing the fate bias predicted by PBA. And what immediately jumps out is that among the early cells, PBA infers that they're bipotent, but in fact, they appear to be relatively committed, yeah?
Yeah, so what happens if you just change your diffusion parameter to make it more, would that make it more faded?
Yeah, so what happens is that if you lower the diffusion, then it creeps up a little, but this is actually the most I could get it to creep up. And what starts to happen at that point is that the progenitor region becomes very salt and pepper. So cells seem to just randomly choose one or the other direction. Yeah. But it's not like there isn't anything to grab onto. So for example, PU2F2 is a transcription factor that has a transcriptional, like an expression boundary that's roughly aligned with the fate boundary. And so it's not like you couldn't, in principle, infer it. It's not just an unstructured blob, but it's just not being inferred.
So is that based on, like, are you saying that it depends on the choice of kernel that you're, like how you define proximity between two cells? Like if you subset your, let's say, genes onto the regulators, then you might be able to just, like, by just looking at this gene and then calculating proximity based on this gene would maybe give you a much better survival rate.
Yeah, definitely. Right, so here, this is sort of, this is the most naive attempt. So we just took all the genes, we just did PCA, and ran the method just as we had done it before. But it's a great point that you could start to now think about with this, now that there is a sort of benchmark, like what would we have to change about the implementation to do better? Yeah. So now in the rest, I'm going to try to see if we can shed some light on the fundamental unknowns and how the individual velocity of cells relates to their average velocity. So I think we've described this ad nauseum. The question is, are there hidden variables? So to figure out if they're hidden variables, I guess, yeah, I want to walk you through how this is going to be set up. So imagine that we're playing a game. So in the game, we took a progenitor cell and let it divide. one sister I'm going to keep, and the other sister, the one on the left, I'm going to give to you. And I'm going to take my cell and put it in my culture dish in my lab, and I'm going to let it differentiate and then check what fate it turned into. Your task is to predict what happened to my cell. You have two options for how to go about this. One option is you can look at your cell immediately, or you can put your cell in an identical culture well in your lab and then look what it did. And you have access to a lot of information. Like, you have access to 1 million iterations of this game that's happened previously. So but what you don't, so there's only one unknown here, which is just what happened to my cell in particular. Now, I want to claim that if there's no hidden variables, you're better off looking immediately. Whereas if there are hidden variables, you may be better off looking later on. If there are no hidden variables, then everything about what the cell is going to do is encoded in the measurable properties of the cell. So if you have access to 1 million previous iterations of this game, then you should just look at what the cell looks like now, then compare it to all the previous iterations, and on that basis, make a prediction. On the other hand, if there are hidden variables, then it might be better to let the cell differentiate, because there might be like chromatin or protein states of the cell that you can't see right now, but they'll eventually manifest themselves in the cell's fate. To make this formal, we can think about the data processing inequality. So so we mentioned before that no hidden variables is equivalent to saying that the dynamics are Markovian. And in particular, it means that so we can think of these 4 cell states, the 2 sisters at the beginning and what they turn into later, as 4 random variables. And to say that there's no hidden variables means that these 4 random variables form like a Markov net. They're like Markovian with respect to this graphical model. And according to the data processing inequality, in this type of graphical model, the information the mutual information between A and D would have to be greater than or equal to the mutual information between B and D. So we're going to call this lateral prediction, predicting D from B, versus forward prediction, which is predicting D from A. And then the claim is that if lateral prediction is better, then there must be hidden variables, because like arguing by contradiction, because if there weren't hidden variables, it would always be better to do forward prediction. The way we can implement this in our experiment is to compare 2 types of clonal relationships that we got. One is the state-fate relationship, and the other is the fate-fate relationship. And just to give an indication, I'm showing you some data from the fate-fate relationship. So what's being shown on the right heat map is so each row is a clone. The left heat map is showing the distribution of fates that clone took on in the left well, and the right version of the heat map is showing what happened in the other well. And what you can see in the correspondence between the 2 heat maps is the fact that cells, clones split into different wells, often did the same thing. So that indicates that cells were relatively committed, actually, at the beginning of the experiment. And when you do a systematic, so we used like machine learning to do a systematic comparison. So we just said, okay, take this sister cell, take the early cell, the state, and then just train a classifier to predict the fate of its sister, and look at the accuracy of that, and then do the same thing, but using the fate of the sister later on to predict the fate of its sister, and that's being shown on the y-axis. So the fact that all of these lines, which are different machine learning methods, fall above the dotted line is an indication that systematically lateral prediction is better than forward prediction. So that's telling us that in this system, there appears to be some hidden variable.
So it seems like missing heredity if you had really bad SNP arrays. Like, in the sense that, suppose what matters, like, when you're done, it's very easy to tell what you are. But at the beginning, this could be at low levels. You're measuring 1% of the molecules. And so, to what extent is it just like, there's of course a missing variable, but that might just be the other 99% of the RNA, as opposed to like one of these outside influences that you like, neighborhood or something like that.
Yeah, definitely. So we have no idea if it's just the unmeasured fraction of the RNA or if it's other things in the cell that are like fundamentally orthogonal to the RNA. And in a way, it's like, well, who cares? Like, because we don't know what to go after at this point. So I guess, yeah, the way I think about the utility of this is it's like missing heritability in population genetics. It's setting up the benchmark, like what's the total amount that we could ever hope to be able to infer, like the underlying encoding of this trait in the genome, and then now we can say like how close are we getting to this? And this is saying that there's some limit beyond which you just can't go. The cell just doesn't know what it's going to do, and so this gives us the gap between what we can currently predict and what's actually there.
But you're undersampling the RNA in the lateral comparison as well as the forward comparison. So I don't think it's just the.
Yeah, so it's not like. Yeah, so this gives us a lower bound for the gap.
Yeah, like undersampling RNA is like measurement noise. This is more like dimensions that you're not seeing, like the proteins or something. Or like if you were to just not measure 500 of the genes, that's different than like measurement noise, or if you had low counts, right?
And there's an identifiability problem in this case between those two outcomes.
And also, would you consider the cell-to-cell interaction, like the communication, as a hidden variable or the model? Because like we are missing a link between the terminal states, that like we assume that they're not interacting. But if there's like equal persons or some other like cytokines which are present in the later state, which would like signal both of them together, then you should add a connection between them, because that would also in other ways extra, right?
Yeah, so it depends on the context. In this case, the extent of the hidden variable is cell autonomous because at the beginning of the experiment, we're taking the two sister cells and putting them into different wells. So that any commitment they have was cell autonomous. But in an embryo, for example, it could very much be dependent on interactions. Yeah?
Have you studied like finite size scaling of this problem in the sense that if I have like only 10 examples of forward, like 100 examples of tap, you have different number of clones. So are we already sort of saturated on the maximum accuracy they can get on the forward prediction? Does it make sense?
Yeah.
Like if I give you a million extra examples, can you make forward prediction better, or are we already there?
That's a great question. So we haven't done a sort of saturation experiment. So the question is, in theory, what we want to be comparing is the perfect map you could ever achieve from our measurement of state in the two scenarios, but we don't actually have a perfect mapping. What we have is sampled pairs from which we're constructing a mapping. And in particular, to do this, we're training a classifier on half the examples and generalizing to the other half of the examples. And we know that that classifier will get better if you just have more training examples. And so the question is, and in particular, these two different predictions, forward prediction and lateral prediction, may be at different points on that saturation curve because of the nature of how we set up the experiment. So, have we ruled out the fact that it's nothing intrinsic about the nature of the regulation, but it's just our failure to saturate the classifier? The answer is I don't really know, but it's roughly.
You have error bars though, right? So you
that's
what those indicate, right?
You could subsample and.
Yeah, that's presumably what he's
Okay,
done,
that's-
right? Because he has error bar.
Yeah, the error bars is we keep shuffling into training and test sets over and over again.
That doesn't give it.
Right.
Because I mean, your sample set is still the same, then shuffle it.
I bet the variability would give you an estimate of how concentrated it is.
Let's come back to it in the discussion.
So, okay, next, diffusion. So this, again, now we're asking another way that two cells can be going in opposite directions despite being in the same state, and that's stochasticity. So the first context in which we're going to look at diffusion is in the context of pseudotime. So here the idea is we're going to take just one trajectory out of this data set and then ask how cells move along this. And the idea is that if there's high diffusion, then cells will be spreading out as they go. There'll be a lot of variability in how fast cells go through pseudotime. Otherwise, it'll be very deterministic. So what's being shown in the middle heat map here is, so I've binned pseudotime into 40 actually I don't remember, maybe it's 20 bins. And what this is showing a joint distribution between the pseudotime position of cells across 2 days. So the heat, the diagonal band is showing that in 2 days, cells generally go forward in pseudotime. And this is giving an indication of how far forward they go in pseudotime. And another thing we can do is look at cells in different wells and ask if 2 cells were sister cells and they're in different wells, how correlated is their pseudotime with each other? So if there's no diffusion at all and cells are completely deterministic, then whatever the starting correlation is between pseudotime should be perfectly maintained over time. On the other hand, if there's a lot of diffusion, we should see that the correlation between the pseudotime of sister cells in different wells deteriorates over time because some just randomly advance farther than the other. So that's what we're testing here. And what you can see, so the 3 scatter plots shown on the right is the relationship of pseudotime for cells in separate wells across on day 2, day 4, and day 6. And what you can see is that the correlation doesn't significantly change. So this is sort of an indication that the diffusion is quite low, at least. So, or at the scale that we're measuring, the stochasticity is pretty minimal. Another surprise that we weren't expecting, but came out of the data set is 2 paths of monocyte differentiation. I'll explain this, and then it will also teach us something about the amount of diffusion. So the idea here is that, so when we looked at the monocytes, so actually spurred by a paper that came out last year that showed more or less the same as what I'm showing here, we wanted to investigate this phenomenon, which is that monocytes come in 2 different flavors. So there's a kind of neutrophil-ish flavor of monocyte, which is being shown on the left column. And so this is showing that the bottom part of this like low-dimensional visualization of the monocytes express neutrophil marker genes. In the next column, I'm showing you the expression of dendritic cell marker genes on the same collection of cells, and you can see that they're more on the upper part. And by the way, in this diagram, differentiation is happening from the left toward the right. So, and then finally, on the right column, I'm showing monocyte marker genes, which are ubiquitously expressed among these cells. So what we did is, this raised a question for us, which is, are the neutrophil-ish monocytes more related to neutrophils than the dendritic cell-ish monocytes? And that's what's being shown with the blue and yellow on the bottom left. On each cell, I'm plotting what fraction of its sisters are neutrophils. And you can see, and so yellow means that that cell is very related, clonally related to neutrophils. And the fact that that's all at the bottom says that somehow these neutrophil-ish monocytes are actually related to neutrophils clonally. And also, similarly, the dendritic-ish neutrophil monocytes are related to dendritic cells. And then we did a state-fate analysis. So here I'm showing on the right that the monocytes just manually divided into 2 categories, the dendritic cell-ish ones and the neutrophil-ish ones. And then the blue and red dots show which cells on day 2 gave rise to those different monocytes. And again, there's some sort of segregation in state space. And putting all this together, we arrive at a picture roughly like this, that the blue cells differentiate into the dendritic-ish monocytes, and at the same time, also, some of them give rise to dendritic cells. And then the red cells give rise to neutrophil-ish monocytes, and at the same time, they give rise to some neutrophils. And the reason this bears on the question of diffusion is that you can imagine if there were a lot of diffusion and stochasticity in the system, at least in the way that we formulated the diffusion with population balance, there's no way you could sort of maintain these 2 parallel streams over time. They would start to mix with each other. So this is a second piece of evidence that the diffusion or stochasticity is quite low. So recapping on everything, population balance helps constrain fate, and it partly recapitulated the FACS results that, and also, but however, it wasn't very good at predicting fate boundaries. So fate commitment, according to lineage tracing, happened much earlier than PBA detected. And regarding the fundamental unknowns, we've shown that at least for this in vitro system, there do, in modulo some of the caveats that you all raised, there appear to be hidden variables. So the cells know what they're going to do better than we can, beyond what we can see, either because we're undersampling or because of it's dependent on chromatin and stuff like that. And then finally, while their hidden variables is a major concern, stochasticity appears to be quite low. So the pseudotime remains correlated over time, and the two paths of monocyte differentiation don't mix very much. So, okay, I think now I'll just be done. Oh. Sorry, also, I just want to thank Alon for mentoring all of this work, and Marav and Sam, who contributed majorly to the PBA project, and then Alejo and his advisor Fernando for their contributions to the lineage tracing, and the MIA organizers. Okay, now I'm done. Thanks.